Well, hello, everybody, and welcome to another edition of Inside the Lab with the CMBC. I'm the director of the center, Dietrich Stout, and joining us today is Robert Liu, who's a professor of biology here at Emory and runs the Computational Neuroethology Lab. And has also just accepted the position to be our associate director. So welcome to the center, Robert, and we look forward to hearing some more about your research today. So I thought maybe I could just lead in for those of us who don't already know, what exactly is computational neuroethology?
Hi, Dietz. Thanks for this opportunity to chat with you and to tell you a little bit more about our research. So to get right to your question, computational neuroethology. I'm going to start off with the neuroethology part. Ethology is the study of natural behaviors. Neuroethology is trying to get at the neural mechanisms of those natural behaviors. And my lab, I called the Computational Neuroethology Lab because in trying to get at those neural mechanisms, it's often much more complicated than traditional laboratory experiments and how to understand how the nervous system actually mediates those behaviors. So you have to often use more complicated, advanced methods to try to get at that. And so that's where the computational aspect comes in. It's a phrase, computational neuroethology, that I kind of came up with when I was a postdoc at UCSF, and it's just sort of stuck. I thought it really characterized my own interests in neuroscience really well, and so I decided to name my lab after that.
Okay, well, thanks. And so you work mostly with rodents, is that correct?
That's right. My lab here at Emory is working with mice and with prairie voles.
Yeah. So, coming from anthropology and so forth, and we have primatologists under the umbrella, and actually a lot of people who study other animals, there's a perception that the reason to study other animals is to understand people. And so is that where you're coming from? But then sometimes I get kickback like, no, I actually just want to understand the animals. So the ethology part of what you're doing, what's the sort of the objective there?
Yeah, great question. Let me step back even a little bit further to explain a little bit why I got into neuroscience. I really wanted to understand something about myself, and which is often what a lot of neuroscientists and scientists do the research that they do. For me, my curiosity was about, why do I not recognize people's faces so well? How do I recognize people's voices, my memory? Those kinds of questions were kind of in the back of my mind as I went into neuroscience. And as I kind of looked around in neuroscience, one of the things I realized is that if you really want to understand how the brain works, you really want to understand it in the context of the ecological environments that that brain has evolved to function in. And so that's, for me, how I got to ethology. I also, coming from more of a physics background, wanted to get more at mechanisms and principles. And so for me, going to an animal model where one can investigate things at either a systems or cellular level was more appealing to me. And so I chose to go into research looking in rodents, where you can sort of access that. Nowadays, I have to say, research in humans has kind of progressed to the point where people are doing those kinds of recordings, but that's not where things were when I first made this transition into neuroscience. And so that's kind of how I came into working with rodents, curiosity about sort of cognitive phenomena that we experience as humans, but trying to understand it at a more mechanistic level that might have general principles that span across different species.
Okay. And so, yeah, so you mentioned that the human research has come along, and it certainly has. Are you interested in turning a bit to work with humans? But surely there are still advantages to working with the rodents as well as drawbacks. And what's that equation look like for you, or for people entering the field now?
Yeah, my impression about the field right now actually is that there is a kind of explosion of both interest in young people going into neuroscience in human studies, um, maybe more so than was possible or that I saw, 10, 15 years ago. And in a large part, that's because techniques have sort of developed to the point where you can start to ask a bit more sophisticated types of questions about how the brain works in humans in certain types of tasks and things. For me, it's interesting you asked because we recently started a collaboration with a couple of faculty here at Emory to try to apply some of the principles that we learned from our mouse studies to see if they might impact human memory and memory consolidation, and whether we could potentially even manipulate that. And so that's been one inroad for me to start actually exploring maybe applications to human understanding the human brain.
Could you give maybe an example of one of those principles, if it's not too
Yeah,
lengthy?
sure. I mean, this for us is a relatively new thing, but a few years ago, in collaboration with Kerry Ressler, when he was here at Emory, we published a study in Neuron on how fear learning in mice, which is a model that's sometimes used to understand neural circuits that may be involved in phenomena like post-traumatic stress disorder, how fear learning of a sound cue that can predict something traumatic like a shock for a mouse, how that involves the sensory cortex, specifically in our case it was auditory cortex, and consolidation mechanisms that occur within the auditory cortex. And so the idea in that study was that by silencing activity within the auditory cortex after animals had learned about a cue being predictive of a shock, a mild shock, if we silence that activity after that experience, that actually seemed to attenuate how much fear they expressed the next day when we tested them with that cue by itself. So it seemed to dial down how much fear they expressed. And so the thinking is that it interfered with the consolidation process for that cue. So taking that study and thinking about how it might be applied in human memory consolidation for cues that are experienced in, say, a traumatic environment, we're thinking that if we might be able to silence sensory cortical activity in a period soon after a traumatic experience, perhaps that could have a benefit for dialing back how that sensory cue gets consolidated in a way that would maladaptively drive post-traumatic stress response. And so that's an area that we're just starting to explore.
So I have to ask, like, how would you silence auditory cortex in a human? Are we talking about some kind of TMS intervention or something else?
Well, that's exactly what we're trying, is a TMS intervention. Yeah.
Okay. So theoretically, that could be clinical if you were able to be there at that moment.
Yes. In theory, and that's exactly what our pilot grant funded by the Emory synergy grant mechanism is trying to test. And so I should mention that's a collaboration with Jenny Stevens and Michael Borsch here at Emory in the School of Medicine.
Cool. And so then, but then more theoretically on that's also very interesting because, I think there's a perception that, perceptual systems are basically input structures to the process that happens centrally. Like, the auditory cortex is just going to feed the sound, and then you have the reward circuitry or the fear circuitry that's going to make associations. But this work is suggesting that it's actively involved in informing those associations.
That's right. And in fact, that's some of the work that we're doing in the lab now in our animal models, looking at how the active processing of sounds as animals are learning about sound behavior associations is engaging the auditory cortex, and how that may be then contributing to how animals learn about that association.
Okay, so kind of a sensory grounding kind of thing.
Yeah. My own way I think about this, I guess, is at any moment in time, there's ongoing activity in your brain. You may be thinking about things. There's unconscious processes going on, but how you interact with the world in a way that then guides what you subsequently do. It begins with the sensations that you have, the cues that alert you to things, or that warn you about things, or that may be pro-social or attractive to you. Those cues out in the world that draw sort of a salient impression will then come to guide your behavior. And so that's what we're interested in, is how those sensory cues that become behaviorally relevant to you come to actually affect how you behave.
Yeah, I find that very appealing way of thinking about cognition as well. I did want to return to something that you mentioned earlier, and that I only really learned from your website, which is that you have a background in physics. So I was wondering, if you could say a word or two about how you wound up training in physics and then going into neuroscience.
Yeah, that's quite a story. I think, in every person's life, in their training, they face these multiple points where they think about what they're doing. They wonder, is this the right path? Is this what I want to be doing? For me, and I think this happens to a lot of graduate students, some point in graduate school when I was studying physics, and doing some very hard experiments at very low temperatures, competing with many different labs, trying to do similar things, thinking, is this what I want to spend my career on as a scientist? Well, is this what I would find myself most curious about? And then realizing that my actual curiosity had more to do with things that were going on in my own head, and wanting to understand sort of the processes that give rise to my ability to sense things, to think things. And so I kind of got interested in neuroscience as a graduate student in physics, and audited a bunch of classes. I talked to other graduate students who I knew were in neuroscience, and discovered that there had been a recent tradition in physicists moving into neuroscience, a very interdisciplinary field at the time. I became aware of work from various theoretical physicists that gave me a sense that there are general principles to learn about the brain that were still to be discovered, and that seemed really exciting to me. And so I made the choice to apply to several institutions that had programs funded by the Sloan Foundation to bring people with more physical sciences background in to work with systems neuroscientists mostly to learn about the brain. I ended up at UCSF as a postdoctoral fellow, working with a number of different people in the Keck Center at UCSF for integrative neuroscience. And that was kind of my my first integration with the neuroscience world.
And do you still find your physics background to be useful to you now?
Absolutely. So I went into physics as an undergraduate and then as a graduate student, I think largely because I thought of it as training for how to think. I also remember this time as an undergraduate student where I was trying to decide between different majors, math and engineering and physics. And math, I had a great teacher. I loved the topic, but was worried it was too abstract. And engineering was kind of the other end, maybe too practical. And I remember having a conversation with my father about what should I choose as a major, and he kind of mentioned, oh, there's all these people who have physics majors or physics degrees who end up in all kinds of jobs, CEOs, data analysts, all sorts of things. And I think it's largely because you kind of learn a certain rigorous way of thinking about problems. And so, you can't lose in some ways going into physics. So that kind of helped me make that decision. And then I always viewed my my training in physics in the same light as kind of a way to think about problems. And so now that kind of rigorous thinking process is what I take with me into every kind of neuroscience question that we look at. Not only the sort of computational and applying kind of computational methods to the questions that we look at, but also in kind of reasoning through the phenomena that we're investigating.
Cool. And just as you were talking, it sounds like maybe your father knew a thing or two about physics. Did he have a physics background?
He was an engineer, actually.
Okay.
So he did not convince me to become an engineer, but yeah.
Cool. And also earlier when you were talking about making this transition, you mentioned that neuroscience, I think you said at the time, is very interdisciplinary. And maybe it's developed more into its own thing now, so it has its identity. But I think there's still a lot of physics in neuroscience. I mean, is there any sort of infusions from physics into neuroscience that you think are particularly interesting now?
Interesting now. Well, where I thought we were going to go was to talk about sort of earlier ways in which, say, modeling has informed neuroscience from as early as just explaining the principles of an action potential, which is something that I teach in my introductory neurobiology class. And that kind of electrophysiology is still at the heart of what my lab does and what I get excited about. I mean, I think at this point, we're in a phase of neuroengineering in many ways, where people are trying to interface computers, hardware with the brain and understand both at a tissue level, how do you get two very different substrates to talk to each other and to live together in some ways without doing damage. And some of that's going to involve physical principles as well as biological reactions and things. And I think there's still a lot of ways in which the two fields can help each other.
Yeah, that makes sense. This sort of integration that you're talking about is very interesting. with augmentation and so forth, a few years ago, people would be talking about, connecting machines to brains and so forth. And I really didn't believe it was going to happen. So we're so far from knowing how the brain works that there's no way that but apparently you can do it even if you don't really know how the brain works, the same way this deep learning stuff works with the language models. So
a little scary, isn't it?
The sort of neuroengineering can proceed even if we're not quite sure what's actually going on, I guess.
Yeah, you're absolutely right. And I think there are pluses and minuses to that for sure. Much of what's being done is being done, I think, for therapeutic reasons, for good cause. And I think we will make a lot of progress on that in the next few years. More non-invasive techniques that might be used for other purposes. I think there will be people who try to do that. And personally, I have my qualms about that, but I think there will be an effort to move in that direction.
I think you're right. So maybe to return more to your own work, you did already talk about some ongoing work that you have, but I was wondering if you could give people just an impression of what it's like to work in your lab. Like, what do you do each day? What do your students do? That kind of thing.
Yeah. I'll say a little bit about what trainees do in the lab first. So we're mainly a physiology lab, which means that we like to put devices in the brains of animals so that we can monitor how the brain is functioning while animals are either doing a task, behaving, listening, or reacting to some stimuli. And so there's experimental work. The nature of physiology, though, is that you collect a lot of data. Some of it you have to winnow out the stuff that's kind of garbage or noise, and so there's a lot of post-processing of neural data that has to be done, and so there are various pipelines for doing that, and then you have to make sense of what you've collected. And the analysis part is, I think, where my own physics background and my own sort of taste for what is interesting, etc. That's where I try to convey that to students and help them think through how to analyze their data, how to make sense of that. Besides physiology, we also study these natural behaviors that we collect. Different types of behavioral data, videos of social behavior of animals interacting socially, or also audio recordings, recordings of the vocalizations that animals make as they're interacting or investigating other individuals, and then synchronizing that with neural activity to be able to look at how the brain is responding to these kinds of cues. So all of that data takes a lot of processing. So there's often, a period of intense data collection, and then a much longer period of data processing and analysis that happens. And then, of course, on top of that, there is, writing papers so that you can get your findings out there, and writing grants to make sure you can keep funding that kind of work.
So that does sound like a vast amount of, especially data processing. Are there sort of existing pipelines for you to do, or is there a lot of just in-house tinkering, figuring out how to get things going?
There are many more tools nowadays than there were 20 years ago, and we take advantage of some things that other people have put out there. There's also our own pipelines that we have developed, or some hybrid of those two. So as an example, in our electrophysiology studies in the auditory cortex of mice, where we use silicon probes with many channels, we take advantage of software to sort individual single units and visualize the quality of those units and where they are within the brain regions that we're recording from. So use some of that software, and then we'll write our own software also to then post-process the outputs of that so that we can, clean it up some more and make sense of how that activity we've recorded relates to the different stimuli we've presented or the different behaviors that are going on at that time.
Nice. And so you already, again, mentioned some ongoing work that you have, but I was wondering if you could, comment on some of the more exciting projects or things that you're interested in that are going on in your lab today.
Yeah, sure. So there's kind of two main areas in my lab. One that's studying, both are studying natural communication, social communication. One part of my lab studies the auditory system and how it processes vocalizations, communication sounds, and learns through social interactions that different types of sounds are meaningful. And the other part of my lab studies, in collaboration with Larry Young's lab at Yerkes, how neural circuits are involved in pair bonding in the monogamous prairie voles. So that we've got some exciting things in both of those stories. Most recently, we published a study that a postdoc did in the lab, where she looked at the neuropeptide oxytocin, which is of huge interest in the social neuroscience community, and is known to play a role in pair bonding and in social behaviors across species. In the prairie vole, one thing that she discovered, looking at a part of the prairie vole brain called the nucleus accumbens, this is kind of a switchboard or a gateway for releasing a lot of motivated behaviors, and it's been implicated in pair bonding in prairie voles, and especially how oxytocin acts on this brain region to facilitate pair bonding. She found that the social experience of interacting with the partner actually changes how oxytocin is acting in the brain. And that's something new. One always just sort of thought that, well, you have these receptors, you have the neuropeptide, it binds, it does something, and just whether or not it's doing it is all we needed to worry about. But her studies suggest that depending on the social history that an individual has had, that actually seems to change the way in which oxytocin functions. So in individuals who have not formed a pair bond yet, she found that the release of oxytocin, which she studied in a brain slice preparation, actually doesn't seem to have a very profound effect on how neurons transmit signals, excitatory signals between each other in the accumbens. However, if animals have gone through pair bonding, that they formed a partnership with their a pair bond with their partner, then when you spritz on oxytocin into the nucleus accumbens, you actually get this what's called a potentiation. You get this increase in how excitatory signals transmit into this accumbens, which then we think plays a role in helping to release affiliative behaviors. And so it seems like that social history can condition the way in which oxytocin acts in the brain, and may explain why in other species and other behaviors, there can be such huge individual variability in the ways in which something like oxytocin actually affects individuals. It could be that the actual underlying cellular mechanisms, molecular mechanisms, have actually been changed by experience.
Wow. So that's really cool. It's also a little bit daunting, because it just speaks to the complexity and the difficulty of generalizing. It's hard to anticipate all of the different things that might be at play in a system like
Absolutely,
that.
yeah. And that actually brings us back to the ethological approach, which is in any model system, it really, I think, is important to think about the ecological context in which that brain has evolved and whether or not the mechanisms you're studying may be adaptive for their behavior within their ecological niches. And those are like when we can understand at that level, that can help us understand what principles might be more generalizable across species and what things might be more specific within a species.
Cool. Well, I really look forward to learning some more about that and talking some more with you now that we're going to be working together at the CMBC. Yeah, before we wrap things up, I don't know if there's anything else that you think might be interesting or important to talk about.
Sure. I guess the other exciting thing from our auditory studies is to look at how the brain, the auditory system is changing as it learns about the exact acoustic structure of sounds that mean something, that mean like, that have a social valence associated with it. And the studies that we're doing now with recordings in animals that are actively learning are going to be, I think, surprising to people because it suggests that there is this active contribution from very early on, even before animals express that they understood that a signal actually means something, that there's already this very active engagement of the auditory system in trying to bias how they behave within a context. That's a little bit vague, so I realize, without spending a lot more time talking about it.
Would you compare that at all to the concept of implicit learning in humans, where people learn patterns and so forth without necessarily being aware that they are?
That might be related, yeah. So that just that exposure, that presence with the stimuli is already leading to some changes within the sensory system itself, some activity within that sensory system that comes to maybe direct how they're going to behave in that context, even though they don't maybe explicitly make an association. And that's actually contributes to some of my other interests outside of our research, which is in the role that implicit biases can have in how we unconsciously make decisions. Sometimes I think about my own research on sort of the predispositions that we have, the biases that we study in our animal models, how that might be reflected in also predispositions that individuals have and that they take into any particular new context as they kind of survey the environment and understand what's going on. And that sometimes can have detrimental effects, but yet it is somehow fundamental to how the brain actually functions in any environment.
Yeah, I think we all like to think of ourselves as like kind of top-down executive creatures. We make decisions and we know how we're going to handle. But in this and also the earlier work you were talking about with the role of auditory cortex and sensory cortex and making fear associations and potential role. There's a lot of this stuff is actually bottom up and outside of your realm of awareness.
And that's a tension, the interaction between bottom up and top down, that I think is really sort of a centerpiece of what draws my curiosity.
Yeah. Cool. That's also something I think we could continue to talk about in the future. I just wanted to say thank you, Robert, for joining us today, and I've enjoyed talking with you, and look forward to continuing our conversations.
Well, thank you very much for this chat, and I really look forward to working with you also on the CMBC.